So now we're going to move on from Chapter 3 to Chapter 4. And before we actually start talking about proteins, I'd like to know if people have any questions. Do you have any questions about Chapter 3 or the material that we covered in it? Any questions? I'll be talking probably on Monday about the procedures we'll be using for the hour exams because we're going to be split between two rooms. But I want to have a written handout for you when I do that so you have something to take away with you so we can get it to go as smoothly as possible. But just be aware of the fact that our first hour exam is a week from Monday. So we finished talking about the nucleic acids, DNA and RNA, they're monomers of the nucleotides, and how they provide structure, the fact that there's a simple repeating pattern in double-stranded DNA, some double-stranded RNAs, that they're great molecules for information, that their structure permits them to perform the function of information transfer and some other things as well. So that's one of the big categories of biological molecules. The next big group of biological molecules we're going to talk about are the proteins, polypeptides. And by mass, they're a bigger part of life than the nucleic acids. They're molecules of a great many different kinds of functions. I think I mentioned during our DNA chapter that we have in the order of 25,000 genes. So there are at least that many proteins doing different kinds of things. We saw in the E. coli thing of the kinds of functions that proteins could perform. And what we're going to see is that with proteins, as in so many things, the structure of the protein is going to reflect and dictate the function of the protein. That there's always going to be this back and forth between what the exact structure is and how it performs its particular function. Biological systems, unlike any other scientific system, have function, something that's good for the organism. We're also going to see how the sequence of a protein gives rise to a structure. In most cases to a structure, occasionally to more than one structure, but that the sequence of the polypeptide is going to be the driving force towards the structure of the protein overall. And we're going to take advantage of two proteins that have been studied very extensively, myoglobin and hemoglobin. These are proteins that have been available as pure proteins for a long time. They are among the first proteins to be crystallized, and we understand much more about them than almost any other, than many other proteins. They're both of them oxygen-binding proteins, but they differ in that in hemoglobin, there is a regulated binding of oxygen. So they give us a chance to look at the same fundamental function of oxygen binding in a regulated and unregulated form. And this picture up here on the upper left is just a picture of a myoglobin molecule, space-filling diagram that's, shows it to you as a smooth surface. I think the next couple of slides we're going to be looking at several examples of protein structures to give you a little bit of a flavor for where we're going, the kind of variety that's possible with proteins, and some of the kinds of functions that they perform. This is an example of insulin, which is a 2-polypeptide hormone. Chymotrypsin, we're going to look at when we get to talking about enzymes. It's a protein that can eat other proteins, a protein designed to degrade other proteins. Here's DNA polymerase. This is a protein we'll be talking about in more detail later on, like chapter 17 or so. And it's a protein that can actually interact with and bind to DNA and help the DNA replicate. Here's an example of a transport protein. You remember a very large chunk of the pie chart for E. coli was transport proteins. This is one example of the kind of protein that can accomplish that. This is a fragment of collagen. They say it's a fragment because the full-length collagen molecule would go down to the basement and up to the next story. And so in contrast to the proteins that we've just talked about that are either enzymes or transport proteins, this is a protein that's really a structural protein itself. Its function is to be a structure. It provides a strength and rigidity and, to some extent, elasticity in things like bones and tendons and so on. Plastocyanin we're going to see later on as part of the electron transport chain in photosynthesis. Here's phosphoglycerate kinase. It's one of the key enzymes in glycolysis. We'll look at it again later on as well. You notice each of these is pointing out when in the future we'll talk about these proteins again. All right. So an amazing variety of protein structures and of proteins accomplishing widely disparate tasks. How is it possible for proteins to do that? Proteins are made up out of 20 amino acids that are incorporated into the polypeptide chain directly. There are a few modified amino acids that are modified later on, but thinking about these 20 amino acids will help us to understand the underlying principles of how a polypeptide is put together and how that polypeptide chain gives rise to protein structure and therefore to protein function. So we're going to look at the amino acids to start getting a feeling for them. At the very least, you need to be able to recognize the names of amino acids and their three-letter codes. You need to know if they're hydrophobic or hydrophilic, how hydrophobic, more or less. You need to know if they're polar or charged. The simplest way to do this is to learn them. We are going to see these structures again and again. So if you want to take the qualitative approach, that will be probably adequate for answering most questions, but the most efficient way to do it and the way that will prepare you best for the subsequent part of the courses is, in fact, to learn the structures. And as I point them out, you're going to see that there isn't that much to learn to master these 20 structures. On this page, what we're looking at are hydrophobic amino acids. You remember we talked about the hydrophobic effect and the idea that things that are principally hydrogen and carbon don't have relatively localized charge and therefore don't make hydrogen bonds to water. They tend to end up together. They don't have an attraction for each other, but they tend to end up together. That's going to be very important in looking at protein structure. These amino acids are the ones that account for that. The hydrophobic effect derives a large fraction of protein structure. And so we can look at these eight hydrophobic amino acids, and we can notice several things about them. They come in different sizes, for instance. So the very smallest of them is alanine. You can see it's basically the amino acid, carboxylic acid, amino group, acid with just a methyl group for a side chain. Alanine, A, sort of the beginning of the hydrophobic amino acids. You can get one that's just a little bit bigger than an alanine and by making valine, where instead of just having a simple methyl group, you have a branch chain there. This has been drawn as a straight chain, but we know that given the tetrahedral character of carbon, it's more like that. So valine V. You could, instead of making a V off there, you could add a phenyl ring to an alanine. That's a phenyl ring. You add a phenyl ring to an alanine, you get a phenylalanine. This is quite a bit bigger, isn't it? And this is now an aromatic side chain. So it's not just hydrophobic, it's also aromatic. Suppose you think about having a branched chain amino acid, but it's a little longer than valine. Well, leucine is a little longer than valine. It's, again, alanine, but with a V moved out by one. So it's a branched chain amino acid, similar to valine, but a little bit bigger, a little bigger. Looking at those four carbons, they can be arranged that way, or they can be arranged a second way, like this. So this is isoleucine. It's an isomer of leucine. Same overall size, slightly different shape. All four of these guys have just simply aliphatic side chains. They're just carbon and hydrogen. There's nothing aromatic about them. Here's one that's a little interesting. This has a sulfur in there, and the sulfur has a methyl off of it, so it's methionine. The thiol, the THIO, that's a sulfur kind of stem for things. Methionine is a fairly hydrophobic side chain, basically a straight chain side chain, but it has a sulfur instead of a carbon there. We're going to find out that can get used for things. And then we have the one with the largest side chain, tryptophan. It's got two fused rings. You can see the aromatic character there. It's got a nitrogen actually inside the ring there. So it is the largest and bulkiest of the hydrophobic side chains. All of these have single letter codes. I'm not going to ask you to memorize them, but you're going to see them in places. A for alanine, V for valine, no problem. Phenylalanine is a F. L for leucine. I for isoleucine, M for methionine. Nothing very exciting there. We're going to use T for something else. We're not going to use T here. So think of tryptophan. I didn't make it up. Tryptophan, W for tryptophan. Or if you want to think of it, it kind of has a W embedded in that fused ring. So W is for tryptophan. And then we come to a very strange hydrophobic amino acid. And this is proline. So here's the P for proline. It's just simple carbons and hydrogens, but it comes around and it makes a fused ring where it comes in the backside of the amine group so that it actually is a closed ring structure. So proline is going to be much less free to do things than most of the other amino acids. It has this ring character to its side chain that's going to limit what it can do. And that's your hydrophobic amino acids. So it's a vocabulary to think about protein structure. Now there are other amino acids which are polar in character. You remember the polarity of a bond comes from electronegativity, hydrogen least, oxygen most, so you can sort of look for the places where that's likely to be happening. And the ones where the side chain has been kind of shadowed a bit are all ones where the side chain has some measure of a polar character. And having that polar character, it means it can interact and make hydrogen bonds. It can interact with water. It's not likely to get tucked into the middle of the protein the way the hydrophobic ones would. Now, cysteine is the second sulfur-containing amino acid. It has a thiol. So a thiol is like an alcohol, but a sulfur hydrogen instead of a carbon, oxygen hydrogen. So here's a thiol. So it's alanine and a thiol. And it's three-letter code is cis or a C. Here's serine. Serine is just alanine with an alcohol. So that's going to obviously have a polar character there, an S for serine, serine. You can also have, take a serine and add a methyl group off of its side, and that's threonine. Serine and threonine are obviously pretty similar to each other. They're both fairly small. They're both alcohol groups, but threonine is a little bit bigger. It's got a little bit different shape. By contrast, you look at tyrosine. Tyrosine has a lot more than just serine. So it's like sticking a phenyl ring in between the methyl group and the alcohol. So this is tyrosine. And tyrosine is kind of a funny side chain because this part of the side chain is somewhat hydrophobic, but the alcohol group is obviously polar. And there are proteins in which the aromatic ring can actually be sort of below the surface of the protein with the alcohol sticking its toe out into the water. We have a couple of amino acids that have amide groups. You remember I asked you to refresh your memory of these groups. Here's a couple of amide groups. So asparagine is simply alanine with an amide group with a carbonyl oxygen there and the amide hydrogen. And then you have glutamine, which has one more carbon in there, but is otherwise the same as that. So again, these two are rather similar to each other. They're going to have similar capacities for hydrogen bonding, but this is a bit bigger. Okay, similar general properties, slightly bigger overall size. There are a number of these that are also going to turn out to be important in metabolism. So glutamine is going to be definitely one of the ones that's going to pop up, but all of these are going to turn out to be interlocked with metabolism along the way. So by learning them now, you're going to have carbon backbones ready at your fingertips to think about metabolism. And then the last of them that has a polar side chain is histidine, which has this heterocyclic ring here with two nitrogens in it. And we're going to talk about its capacity to hydrogen bond in a little bit and even to be protonated or ionized. But it's basically just alanine with that ring added on. Then up here in this corner, without any shading on it, is glycine. And glycine is an amino acid with just two hydrogens hanging off of it. So it's the smallest and simplest conceivable amino acid you can make. It happens to be sweet. That's why it's called glycine. It tastes a little sweet if you eat it. But it's not particularly polar. It's not particularly hydrophobic. And it's very small. So it can fit into places that other amino acids can't fit into. And then we have a few amino acids that, in fact, have charged side chains under most conditions, under the typical conditions of a living cell. So we have aspartate. Remember asparagine, aspartate. So aspartate is the carboxylic acid instead of the amide. So you have alanine plus a carboxylic acid. Glutamate instead of glutamine is just one carbon longer, just like glutamine was, and a carboxylic acid. Now we used A for alanine, so we can't use A over again. So D is aspartate, as if you came from the Bronx, aspartate. Glutamate, we've used G for glycine, so we can't use G over again. Think gluey. Things made out of protein get gluey. Again, I didn't make up the mnemonic, but there it is. Now you'll never forget it. And then we have two that are generally positively charged at physiological pHs that have basic side chains. So these two have acid side chains. These are basic side chains. One is just a simple hydrocarbon chain with a primary amine on the end, and that's lysine. We already used lysine, L for leucine, so lysine is K as a contrast. Leucine and lysine sound kind of similar, but they are rather different. One is a hydrophobic one. One is a positively charged side chain. And then the last is one of the bigger ones, arginine, and it has this complicated 3-nitrogen guanidino group here that actually has a bit of aromatic character to it. So that's it. That's all there are. They're built off of each other, so you can learn them by learning one after the other. A lot of people find flashcards helpful for this, and the first few times you go through it, you might want to redraw it. Get one of those dry erase boards and start by drawing a carbon, amino acid backbone, make alanine, then turn it into valine, erase that part, turn it into phenylalanine. Manipulating it with your hands will help to fix it in your head. Any questions about those before we start using them for something as opposed to talking about them? These amino acids in isolation are just small molecules. You find them in deep sea vents. You find them in space. But they aren't by themselves going to do a great deal. One of the interesting properties they have is that most of them, 19 of them, are what we call chiral. And a chiral compound is a compound that comes in at least a left and a right-handed version. So here we have a picture of alanine showing it in two versions. And if something is chiral, you can't superimpose the left hand over the right hand and have an identical match. I have to put my palms together to superimpose them. They're never going to be identical. You can't wear a right glove on a left hand, right? Glycine, on the other hand, is not chiral. Why would that be? Two hydrogens there. So you haven't got any way to tell those two hydrogens apart. Glycine is a mitten. You can wear a mitten on either hand, right? So most amino acids are like gloves, but glycine is like a mitten. And this turns out to be very, very useful in building protein structure. Now, some of these side chains have interesting properties that make them especially powerful candidates for functions inside of proteins. So we looked at initially at histidine in its unprotonated form. Here is as the base form that can become protonated. If it accepts a proton, here it is charged, positively charged. It can donate that hydrogen to something else. The pKa for this reaction is around 6. So when the pH of the solution is 6, half of the molecules will be in this shape, half of the molecules will be in that shape. If you move up to pH 7, at pH 7 there's only 1/10 as many protons in solution. And so you're going to get a shift in how many of these two forms you have. If there are fewer protons in solution, you're going to tend to lose the protonated form and have more of the unprotonated form. So at pH 7, you'd have more of this form, about 90 to 10. So how much you have of it will shift as you go above and below the pKa. That makes it very helpful in enzymes. And we're going to see histidine many times. Another reason to learn it. Another side chain that can become ionized is cysteine. And this is showing you the thiol, the sulfur alcohol, so to speak, the thiol of cysteine. It can lose that proton and become an S minus, or it can pick, the S minus can pick one up and become SH again. Its pKa is around 8. It's roughly a pH unit above neutrality. So at pH 8.3, it'd be half and half this. As you go down closer to neutrality, more of it will be in the protonated form. Again, useful in enzymes. The other cute thing that cysteine can do is make a disulfide bond. So two cysteines under the right conditions with the assistance of the cell can actually make a bond to each other. So you have a sulfur-sulfur bond. And that's going to turn out to be useful for extracellular proteins. I do not expect you to memorize these pKa's, but you should have a general sense of where they lie. Okay? We have talked about two amino acids that have carboxylic acid side chains, aspartate and glutamate, aspartic acid, glutamic acid. Their pKa's are around 4, a bit below, above and a bit below 4. They have acid pKa's. Ah, surprise. About 3 pH units below neutrality. We talked about two that were bases, lysine and arginine, and they have quite high pKa's, actually rather different ones when you look at that. Lysine around 10, so about 3 pH units above neutrality, and arginine 3 pH units higher still. That resonant character in the arginine side chain makes it an extremely high pKa. And then the three others that can be protonated or unprotonated are closer to neutrality. Tyrosine, in fact, has a pK similar to lysine, but it doesn't find itself in circumstances where that's used as often as lysine. Okay. This is just reminding you where those ionizable groups are on these things. Keep in mind that a protein has a carboxy terminus and an amino terminus, just like the amino acid has an amino group and an acid group. So the carboxylic acid at the end of a polypeptide chain also has a somewhat acid pKa. We just talked about aspartate and where those protons go. Here's the histine, cysteine. Now the amino terminus can become protonated into a primary amine unless it's modified. We'll talk a little bit about that later on. It's not quite as basic a group as the lysine would be, and so on. So this is just reminding you of what those structures look like, where the protons come and go from. So 20 amino acids, just by themselves, make up a mixture that's kind of has an odd taste, but it won't do anything very interesting. It's just a solution. You've got to put the monomers of the amino acids together into a polymer to get anything interesting out of them. And in order to make that polymer, you're basically going to remove a water molecule between each pair that's being added together. The exact mechanism of it we'll talk about more at the end of the course, but just in terms of general chemistry terms, what you're doing is taking away a water molecule. So if we just for a moment take these two as examples of amino acids with their side chains. So R is just, this is any side chain, right? So R1, any side chain, R2, any side chain. And what you're going to do is the carboxylic acid is going to give up an oxygen. The amine group is going to give up 2 hydrogens. And so that takes out a water molecule worth of stuff. And what you end up with then is a new thing, a peptide bond. The peptide bond has a lot of really neat characteristics. The peptide bond makes life possible. So you have this carbonyl oxygen with that carbon then attached to the nitrogen, still with this hydrogen off there, and then on to what's called the alpha carbon of the second residue. So this is the center carbon of the amino acids is the alpha carbon, and the peptide bond is formed between what was the carboxylic acid and the amine group. That peptide bond has a tendency to be planar. It's not perfectly planar, but it tends to stay as a planar thing. We're going to look at why that is. And then the R groups, the side chains of the residues, are going to stick out from this backbone. So you can add together many, many amino acids together to make a polypeptide. You have a long chain, and the R groups will stick out from the sides of that. I do encourage you to take a look at the CD. It has a very nice development of that. And I will include some links on Icon to web-based versions of that you can look to if you don't want to use the CD. And this is just showing you that for more than one. So you've got an amino terminus that's still a free amino acid, a carboxy terminus that's still the carboxylic acid, and then in between every was an amino acid, now is a residue, you have a peptide bond. There's one and one and one, with R groups hanging off. Now, the reason that the peptide bond has a tendency to be planar is that it has some resonant character. We're going to run into resonance in a number of places, so this is probably a good place to remind you of what it's like. You could draw the peptide bond in two ways. You can draw it either with the carbonyl oxygen and the nitrogen like that, or you can separate the charge a bit where the oxygen, it has the negative charge, and the nitrogen with a positive charge, and a double bond now between the carbon and the nitrogen rather than between the carbon and the oxygen. Because those are both somewhat equivalent forms, the in real life character is sort of a resonant combination of both of them. And so that bond has somewhat of the planar character, some of the planar character of an actual double bond. And so that means that when you look at a polypeptide chain, the peptide bond parts are kind of like blocks that you're going to work with. You can rotate around the alpha carbon bonds. So here's an alpha carbon bond between a nitrogen and the alpha carbon. That bond will rotate. There's a bond between the alpha carbon and the carbonyl carbon. That will rotate. But the peptide bond will behave more or less as a sheet moving as a body. Now, because the polypeptide has the carbonyl oxygen, sprinkled along every third spot, the freedom to move around this alpha carbon is not absolute. So you've got the alpha carbon here, that's the R side chain there. Here's the carbonyl oxygens. They're kind of big. And so when the polypeptide chain is moving around and exploring space, it doesn't have complete freedom of where it's going to go. There's a bit of constraint of its movements. So now I hope you're starting to have this image in your head of this polypeptide, this long molecule of variable length, depending on what it is, but a linear molecule in which the backbone is a repeating polypeptide backbone, and then hanging off of it are these R groups. And the R groups can be any of 19 different R groups, or 20 if you want to call hydrogen an R group. This allows us to think now about structure at 4 different levels. And although this terminology was developed originally for protein structure, I think you can see the analogies to some extent between protein structure and other kinds of structure that we have talked about and will talk about. Primary structure is the same as primary sequence. So the literal sequence of amino acid residues in the polypeptide is its sequence. That's its primary structure, the first level of structure that it has. And you can specify that by just specifying, amino acid, residue. It turns out to be the case that many polypeptides, given the freedom of movement to live and everything, will spontaneously start to take on more structure than that. And it is often the case, although not always the case, that the polypeptide will take on patches of order over a short region. There'll be a local repeating structure of some kind. That local order is called secondary structure. And there are 2 kinds of secondary structure that we see very frequently that account for a large fraction of the secondary structure that proteins have. And that's alpha helix and beta sheet. And we're going to talk about both of those secondary structures in a little more detail. Overall, when you take that polypeptide and you let it fold up completely in space, so it's achieved its mature folded structure, that's its tertiary structure. That's the 3-dimensional packing of the entire polypeptide chain. And many proteins have a particular tertiary structure. We're learning that there are some proteins that are a bit floppy unless they're binding to something else. But they have a tertiary structure that they will take on when they're fully functioning. So tertiary structure is all the packing of that complete polypeptide chain. Now, many proteins consist of only one polypeptide chain. So they have primary, secondary, and tertiary structure. But some of the most interesting and essential proteins in life have more than one polypeptide chain. They might be dimers or trimers or tetramers and so on. Hemoglobin is a tetramer. And the way in which those polypeptide chains pack together to make the tetramer, that's quaternary structure. So quaternary structure is only going to be present if you have more than one polypeptide chain. Any questions about this before we talk about some of these in a little more detail? And remember, Eli, you're supposed to flag me if I get egregiously wrong, right? He works in a structure lab, so if I make mistakes, he'll find it. I should actually say that's a broad invitation. If you hear me making a mistake, I mean, I am human. I can make mistakes. If you hear me making a mistake, please flag me down at the time. I would much rather correct it now in front of everybody than try to do it after the fact. So if you hear me make a mistake, please do catch my attention. So let's look at secondary structure. We've talked about primary sequence or primary structure, and we'll talk more about that in a little bit. The fact that you have this repeating polypeptide backbone makes certain things possible. We mentioned already that the planar peptide bond is not perfectly free to rotate. You can imagine that the side chains, the R groups, are different in size and so on, that the big ones are not going to fit into the same spaces as small ones and so on. So that's going to be a limit to how things pack. But what's quite interesting is that the carbonyl oxygen of the amide bond is potentially a hydrogen bond recipient, and the hydrogen off the amide bond is potentially a hydrogen bond donor. And so what can happen is you form hydrogen bonds between parts of the polypeptide backbone. And this is an example of how that can play out. This is what's called a ribbon diagram of the alpha helix. And you can see here hydrogen bonds that are going up and down that helical structure. So here's a hydrogen bond, here's a hydrogen bond, here's a hydrogen bond. So as that polypeptide backbone is twisting around to make the helix, there are hydrogen bonds running up and down that helix, all up and down it, like the bark on a tree almost. This is a helix, by the way. This is not a spiral. Spirals are flat. This is a helix. Helix goes up and down, just so I don't see any spirals. So this is one of the two common ways that secondary structures can form. And it will be something that reflects sort of the minimum strain for that particular sequence of polypeptide. Different sequence of polypeptide, different secondary structure, depending. So let's think a little bit more about the alpha helix. The alpha helix was the first secondary structure that we really understood. The polypeptide backbone makes a coil or a helix going, turning. It's a right-handed helix. You remember that DNA, A and B DNA were both right-handed helices. Here's another right-handed helix. There are about 3.5 residues per turn, so about 3.5 side chains sticking out per turn. It takes, the distance between a repeat is about 5.5 angstroms tall. And I mentioned that the hydrogen bonds are running up and down the long axis of the helix. Now, something that you don't, this one makes it a little easier to see the polypeptide backbone, that it's coiled going up in that helix. Here's a space-filling diagram, and it makes the point that the center of that helix is essentially solid. It basically fills the space there. And then this space-filling diagram makes very clear how the side chains stick out from that central core of the polypeptide helix. Excuse me. I have a sneeze. Okay. Any questions so far? All right. The second secondary structure that we'll talk about that is also very abundant is the beta pleated sheet. So alpha helix because it was the first structure described, beta pleated sheet because it was the second structure described. In the beta sheet, the polypeptide backbone is more extended. We kind of coiled the backbone in the alpha helix, kind of like an old-fashioned telephone cord. You have to imagine pulling on that to extend it to get a sense of what the polypeptide backbone is doing in a beta pleated sheet. And so here you can see we've pulled it out a little bit, but again, it's going to be stabilized by hydrogen bonds between a carbonyl oxygen and the hydrogen off the amide nitrogen. So the potential hydrogen bond donors and recipients of the peptide bond are the things that stabilize that secondary structure. Now you're going to make a sheet of stuff. You can have 2, 3, 5, a whole bunch of these peptide chains lying more or less side by side. And now the side chains are going to stick out above and below the plane of the secondary structure. You also have a potential in beta sheet that is not there for the alpha helix, which is you have to ask, are the peptide backbones parallel? Are they lined up with their amino terminus at the same side and their carboxy terminus at the same side? Or are they lined up anti-parallel, so that the carboxy terminus is with the amino terminus here and vice versa? Okay? And this is just showing you what parallel beta-pleated sheets do for their hydrogen bonds compared to an anti-parallel beta-pleated sheet over here. Okay? Any questions? All right. And here we're again using the ball and stick and the space filling. If in order to have parallel beta-pleated sheets, you have to somehow or other loop around. The simplest way to make it is anti-parallel, where you come around and just go right by yourself another time. Both for beta-pleated sheets and for alpha helices, there may be loops and things like that, some kind of irregular structure that connects regions of secondary structure. You might have an alpha helix and a little loop, and then some beta-pleated sheet for a couple, and then some more alpha helix. You can mix them up in one protein. This highlights, I think, again, how you have the sheet-like structure in the center and how the side chains are sticking up above and below the plane of the beta-pleated sheet. Okay. So we've talked about primary structure, which is the sequence, secondary structure, which is this repeating local order. Either the most common ones are the helix or the sheet. But native proteins have generally an overall packing in space. And certainly by the time you can crystallize a protein, in a protein crystal, in each individual molecule, most of the atoms have a position. They're somewhere, some parts of it may be floppy, but overall there's a position for most of them. Some proteins in that 3D packing are fairly compact or globular. There are other families of proteins where the whole point of the protein is to make a long, extended, fibrous kind of thing. And the way in which we find that out, there's a couple of methods that are used, but the one that was used first and is still used widely is through things like X-ray crystallography. So if you have a crystal of protein, so here is an actual crystal of protein. It's pure protein, and it has settled down into a repeating pattern so that every protein molecule is sitting in the same orientation relative to the next one. If you shoot an X-ray beam through that, it will scatter in a pattern that reflects the positions of the atoms in that molecule. And so if you look at this X-ray diffraction pattern, you can see that there are many, many, many dots. And by measuring position and their relative position and stuff, it's possible to reconstruct the tertiary structure of the protein it came from. It's also possible to probe it by NMR, which is nuclear magnetic resonance. That takes advantage of magnetic fields and orienting the atoms and then letting them relax. It's a different approach to it, but it has its own strengths. When we are looking at tertiary structure through X-ray crystallography, the exact degree of resolution will depend on the quality of the crystals and the size of the protein, various things like that. If we have a modest degree of resolution at 5 angstroms, say, we'll have an overall sense of where the polypeptide backbone is, but not a lot of detail. And as we get more and more resolution, so down here we're down to 1.5 angstroms, we can start to actually image the individual side chains off the residues. Sometimes now we use synchrotron radiation to do crystallography. So it can get very high resolution as well. There are some other general features to tertiary structure that hold true in general. For soluble proteins, for basically proteins that aren't part of the membrane, in general what we're going to find is that the hydrophobic residues will tend to be found on the interior, and the hydrophilic groups will tend to expose their side chains on the surface. may not be possible to keep all those hydrophobic groups completely buried, but they're going to tend to take on, the protein as a whole will tend to take on a configuration that buries as much of the hydrophobic groups as they can. Although there are certainly some proteins where the polypeptide backbone is all there is, and that's enough to do what it's going to do, there are many proteins where some additional chemistry is needed, that the particular chemistry of the reaction that's needed, nifty as the 20 amino acids are, is not available in that particular toolkit, in which case the protein may have a prosthetic group. So a prosthetic group is something that's not part of the protein backbone, but it helps it. Have you ever heard the phrase prosthesis? Someone who has an artificial leg has a prosthesis. A prosthetic group is a group that helps the protein do something. And this is just a picture showing you how the heme group is the prosthetic group for myoglobin. So if this is the myoglobin structure, here you can see in the bare backbone thing that in here there's a thing that doesn't really look like a polypeptide backbone, and it's not. It's the heme group. It's the prosthetic group. And here the ribbon diagram is showing you the various alpha helices, and you can see how those alpha helices are packing together to make a little pocket for that heme group, for the myoglobin. Okay. Myoglobin is a fairly small protein. It's compact. It's all tucked in together. Hemoglobin is going to be 4 of those packed together. But sometimes you'll get proteins which get larger not by adding several small things, but by having several what we call domains, where one part of it seems to have kind of a compact tucked togetherness, and then there's something loose and floppy that connects it to another thing. And so this is an example of a protein that has separate domains. So this would be considered one domain of it, and this is another domain of it. So the tertiary structure would be the whole thing altogether, but this particular tertiary structure has 2 domains. And in proteins like this, it's not uncommon, it's not universal, but it's not uncommon, that you can clip it in between those 2 things, say with a protease, and that this domain will still do what it does, and that domain will still do what it does. The ability of these domains to fold up compactly as independent domains is evolutionarily very important. It's part of what makes it possible for organisms to kind of mix and match domains to generate new proteins. That you can, translocate a gene to put this domain next to something else, and those 2 things together now become a single protein which can do something that they couldn't when they were separate domains. There may also be interactions between those 2 domains, so you might find that there are side groups, side chains that are interacting along that interface. I think we'll call that it till Friday.